we are totally not getting through this in 20 minutes, even though we only have, 10 slides left, but, okay. So, I'm just gonna go until class is over. We'll pick up next time. Okay? So, we were talking, this is like last Monday now, about muscles. And so, just as a quick reminder, right, we have all of our muscles, and they are composed of these individual muscle fibers. So if we actually look into a muscle, we see tons of these little fibers. They all come together to create our muscle, right? These fibers themselves are the individual muscle cells. So there's this kind of elongated, interesting cells. And they're innervated by motor neurons. And we kind of briefly said, okay, there's going to be two types that we're going to talk about. The main type that's really doing the work of causing contraction are these alpha motor neurons, all right? So they synapse onto the muscle fibers, they send the electrical impulses, and then through a process that's many, many, many steps, the action potential from this neuron gets turned into calcium being released inside the muscle cell. And if you know about actin and myosin and tropomyosin and those things that you may learn about in physiology or something, that's what causes contraction. But ultimately it is initiated by the neural signal coming from the alpha motor neuron. And we said that there's motor units. That would be a single alpha motor neuron and every muscle fiber that it innervates. We also have motor neuron pools. So if you take like your bicep muscle, right, there's several alpha motor neurons that collectively will synapse onto all of the individual muscle fibers within that muscle, and those are the motor neuron pools, okay? And so we said that if you want to have like a small contraction, you could have just a few of those motor neuron pools being activated. But you want a really strong one, you want the entire, all of the strength of that muscle to be working. We're likely recruiting additional motor units, additional motor neurons, and all of those fibers to all collectively contract. And generally that's done from the smallest, for like little fine movements, it's just the smallest motor units, to the largest, getting everybody on board for the contraction. The other thing is the firing rate of the motor units can differ, so that's what this diagram is really showing. If we have motor neuron activity, action potential here, action potential there, right? That we can see how that relates to muscle contraction. Get a little bit of a contraction, but the other one is not close enough that this one has already come down from the contraction before the next one fires off, and so we're kind of getting these little twitches. If we all of a sudden put those closer together, and even much closer together, now we have kind of an additive effect, and we can also have sustained muscle contraction. So that's what's happening when, like I said, your whole, all the strength of the muscle is being, utilized to, contract. Maybe it's to move something really heavy. That would be really fast action potentials setting us up for that. Okay, so we have a couple different types of motor units, actually 3 specifically. This relates to something called white fibers and red fibers, and these are muscle fibers, okay? This is the same idea, it's on Thanksgiving, do you want light meat or dark meat, right? We know that there's a difference between different parts of the protein that's there, okay? The darker, or what we would call in this case red fibers, it's because there's more mitochondria, there's additional enzymes and things present that give it that color. And these are our red muscle fibers, and they are slow motor units. Okay, so these are shown down here. In this case, it's in green and shown by this part of the diagram. But you can see that these motor units, they don't generate a ton of force. Okay, so we're here from 0 to 4 grams compared to up here we're going to get towards 50. So they don't generate as much force, and they're a bit slower to contract. But because they have all the mitochondria and those additional enzymes present, they're slowly fatiguing. So they can maintain that level of contraction here you see time in minutes, for quite a long time without fatiguing. Okay, that's in contrast to our white fibers, which don't have the same properties of being able to resist fatigue for so long. Okay, the white fibers come in two flavors. So we have fatigue-resistant fast fibers. All right, that's in blue here. These ones are much faster to respond, to generate the contraction in response to the neural signals. And they're not, quite as robust in terms of, being resistant to fatigue, but they have some ability to kind of resist fatigue. They're kind of a middle fiber, to be honest. All right, then we have our fast fatigable fibers. And these are our fastest, strongest fibers. They respond really quickly to neural signals, and they do so with a large amount of force. So those contractions generate a large amount of force, but they're very quickly fatigued. So you can only maintain that level of contraction with these fibers for a short period of time before those fibers get fatigued and they need to relax and kind of regenerate energy, kind of like that. Yeah.
Is this the same thing as when people say like fast twitch muscle fibers?
Yes,
slow twitch?
it is related to that. Yeah. Yeah, it's actually the same thing. We're just in this case a little bit more interested in the neural impulses than the contraction properties, sort of, but it's the same idea. Okay, if we were to look in a muscle, we usually see that all three types coexist, but within a motor unit, it's one or the other, right? So you might have a few motor units of these fast fatigable fibers, and then in the same muscle, you have other motor units that are the slow motor units. And that gives our muscles, right? I can, hold something, pretty light in my arm, my biceps contracted for quite a while, but if it's really heavy and I need to use those fast fatigue resistant or fast fatigable fibers, can't hold it as long. So that's all within the same muscle, but there's different motor units, okay? The other thing that's really interesting is that these have corresponding axons that sort of respond to them. And so what I mean by that is our slow muscles are kind of innervated by slow neurons. And these slow neurons are going to be generating these kind of, paced but not so fast action potentials. If we were to go on the opposite end and look at the fast fatigable fibers, these fast fibers are kind of innervated by sort of a corresponding fast neuron, and they send really quick bursts of action potentials, but they, it's just a burst. It's not kind of a continued, release of those quick action potentials. And so that kind of alludes to the question, right? Well, which part, the muscle or the neuron, is truly imparting those qualities of fast or slow? Is it that it's just the neuron and the characteristics of the neuron that then lead to the muscle being a fast muscle, or is it something about the muscle that's feeding back and telling the neuron how to fire, right? There's some sort of synergy going on between those two there. Additionally, it could be like perhaps they're both, encoded to be that way. Fast muscle fibers are genetically encoded to operate that way. Fast neurons are genetically encoded to operate that way. And somehow during development, the two meet up so that they meet. Right? And so that's the question that this experiment was trying to answer, saying what determines muscle characteristics? Normally, we have these slow alpha motor neurons that have these paced action potentials that are continuous, synapsing onto our slow muscle fibers. And then we have the opposite. We have the fast ones, the really quick bursts of action potentials. And then we end up with the properties of those muscles being fast. And so what they did this crossed innervation experiment. So they switched those connections. They said, what if we take a slow alpha motor neuron and we synapse it onto a muscle that is generally behaving as a fast muscle fiber, and then the opposite, fast onto slow. And they actually were able to show that changing the innervation of these muscles actually kind of reversed the phenotype in the muscle. So when we all of a sudden gave the fast muscle fibers the slow paced action potentials, the actual physical characteristics of that muscle changed to be a slow-like sort of phenotype, the actual characteristics that that muscle presents, and vice versa. When the neurons that were synapsing onto our slow muscle fibers were doing the really quick bursts of action potentials, these muscles kind of switched over to have the physical characteristics of fast muscle. And so it's kind of an interesting thought, the idea that our experiences and how that translates to neuronal output, and how those neurons synapse onto these muscles actually can kind of change their physical characteristics. So that's kind of just something interesting to think about. We're going to revisit that idea because there's thought that this plays a role in learning and memory. So the fact that we can kind of change how, if our experiences change our neural output, then somehow that can lead to our concept of learning and memory. Okay, so we've talked about the muscle, the alpha motor neurons, but we need to introduce those gamma motor neurons. And so what we have kind of buried within our muscles are structures called muscle spindles. And these are providing feedback about muscle state. So it's basically kind of a protective thing. We're going to talk about something called the stretch reflex that ensures our muscles are not stretched or contracted beyond their, beyond something that could cause damage. All right, and so inside of our muscles, we have these little muscle spindles, and what they are specialized muscle fibers. So these little orange or yellowish structures are muscle fibers. They're a different type, but they're a specialized muscle fiber in here. And wrapped around it are these group 1A axons. So if you remember, those axons are the ones of the largest diameter, meaning they are really quick to send signals out. All right? And then the whole structure is kind of embedded in this fibrous capsule that's embedded inside the muscle. Okay? And so this is also, we could call this a stretch receptor. Once again, it's kind of monitoring how much the muscle's being stretched. And this is also an example of proprioception, the idea that we have structures on our body that help us kind of, our brain understand the position of our limbs and joints and muscles in space. So this is an example of one. Okay, what these do is, in the stretch reflex, we're going to see that when you have a muscle that's kind of abruptly, there's a quick change in rate of stretch, or there's kind of a quick stimulus that causes a muscle stretch, they initiate a reflex that, upon stretch, causes the muscle to contract a little bit. All right, and so up here we can see our muscle. In the middle is the muscle spindle. So there's these yellow muscle fibers wrapped around the axon. They extend projections out to the spinal cord, where they actually synapse onto our alpha motor neurons, and that's shown in green there. Okay, here we're kind of at a neutral state. What happens if we add a weight to our muscle? So adding a weight to our muscle would be equivalent to stretching it. So you would experience a quick stretch, all right? And so all of a sudden we get an increased load, and then really quickly that causes this elongation of the muscle. We stretch the muscle, all right? We don't want our muscle to just, be able to kind of stretch, indefinitely. And so what happens is the muscle fibers inside the muscle spindle also get stretched. And as they get stretched, those group 1A axons wrapped around them get triggered to send out action potentials. So that's what we see here. In response to the stretch, now this neuron here is sending out quick action potentials to the alpha motor neuron, all right? There's a teeny delay, just the time that it takes for the signal to travel to the alpha motor neuron, but now the alpha motor neuron discharges really quick. Well, what's that doing? It's synapsing onto the muscle, so it causes contraction. And so that's what a stretch reflex is. It's in response to kind of a quick change in stretch of the muscle. It initiates the reverse reaction, right? If we're being stretched, it's going to kind of cause our muscles to pull up really quickly. All right? Yes?
So when you pull a muscle, is it because you overwhelmed your stretch reflex?
I think with that it's more about like we've caused some sort of muscle damage. I'm not sure how much it relates to just the muscle spindle. Yeah. All right, so an example of this is the knee jerk reflex. So you've probably all had the experience, you've gone to the doctor and they're like hitting your knees and you're like, what are you doing? Well, they're actually testing to see that these motor neuron kind of circuits are active and working correctly. And so, there's all sorts of different reflexes. In here, this is the patellar reflex. If you use a reflex hammer and you kind of strike the tendon that's right here, not super hard, but enough to cause a little bit of stretch in the muscle spindle embedded deep in that tissue also stretches. That causes our axon, our group 1A axon, to send the signals out really quickly to the alpha motor neuron, which comes back and tells the muscle, the quad, to contract a little bit. And that's why when your doctor does that, your foot kicks out a little bit. Okay? Yes. Oh, fly. Yeah. And we have similar reflexes in other parts of the body, right? So if you were in neuro lab, we tested some of them, right? You can do it on your tricep, you can do bicep reflex, you can do your Achilles reflex, and it's all the same idea. We're testing to see that these neural circuits are actually kind of intact and functioning. And if you don't have this response, there's a sign that something is going wrong, probably in your spinal cord. Question?
So just to clarify, were you saying that because the tendon was stretched,
Yes.
then the signal to the brain was to contract?
So it's not to the brain. It's actually an important part of this. Nothing's going up to the brain. This entire reflex is mediated solely within the spinal cord. But what you said is correct. When you press right here, I know it doesn't seem like much, but it causes this whole muscle to stretch a little bit. Very, imperceptible stretch to you. You don't feel like your muscle's being stretched, but it's just enough if you press on the tendon that it kind of pulls everything very transiently, and then our spindle gets stretched a little bit, and that signal is sent out to have contraction. All right, and if you remember doing this, or you remember from lab last week, this is very quick, right? You get struck, and the reaction of your knee, your leg kicking out is a very quick reaction. And so that's an example of muscle spindles and this reflex kind of in action. Okay, we are out of time. So where we're going next is we're going to talk about those gamma motor neurons, and then we will next class discuss how does the brain play a role in this input, right? Everything we've discussed so far is mediated solely in the spinal cord, but our brain does send some information down to give us some extra input there. All right, real quick, don't forget, if you want one of those, please grab one of those for the exam corrections. Also, I